In this video, I'm going to show you how I use simulations in my classroom. At johndarko.com, I've created dozens of simulations that I use in my biology and ecology classroom. These simulations are computational based, so there's math behind them, and I do it to help build the quantitative skills of my students, but also really help them pick out patterns that emerge with different dynamic systems and allow them to think about the practice of science. So testing hypotheses and just doing general discovery-based science. This builds off of the constructivist idea that students, by playing with a system, can start constructing their own knowledge. One of the first ways I have my students use a simulation is, before I even run a simulation, I'll have them in whiteboards or in their notebooks draw a behavior over time graph. So just a simple graph like this, and then have them draw how they think the system will behave over time with some type of variable. In this video, I'm going to focus on my photosynthesis model. And so one thing I'll ask students is, how will the amount of oxygen in a leaf change over time? And have them graph that out. I'll then run the simulation, and then we can talk about discrepancies between their mental model and my computational model. Caleb Bryce wrote a nice article for the American Biology Teacher where he talked about the types of models used in science. I adapted that slightly. Concrete model, structure of DNA. You have a computational model like the logistic growth model, and then a conceptual model like the formula for photosynthesis or cellular respiration. I adapted that to also include experimental models. As teachers, we use that as a heuristic. We use experiments to help students both practice science, but also discover something about the science, hopefully. And we choose different model experiments. We use like the leaf disc lab, for example, or we measure the amount of dissolved oxygen in algae undergoing photosynthesis. These types of heuristics or effective uses of simulations. A good review article comes from the Journal of Teaching and Learning with Technology, and this is, they go through several of those, and I've adapted those for this video. Let's take a look. This is an output from running one of my simulations, my photosynthesis simulation, and this is the amount of ATP as it changes over time, NADPH as it changes over time, oxygen changing, and also glucose. What I'll have my students do is look at a conceptual model of photosynthesis, and I'll say, okay, this is the light reaction. What part of the simulation is showing the light reaction? So let's look at my model. I'm going to reset this, run the model. And as I run this, I say, okay, so what part of this is showing the light reaction? What they can do then is look at this conceptual model and compare it with the computational model, and so we get these different forms of representation. What we see is in the light reaction that oxygen gets produced, NADPH gets produced, and ATP gets produced. Well, when we click on here, we get different readouts, and we can see that in this stretch of time, from time interval 3 to about 14, ATP and NADPH go up, and this is showing the light reaction. Then the Calvin cycle with glucose getting produced, and then the light reaction, and then glucose gets produced, and then another light reaction. So there's kind of 3 cycles of the light-dependent reaction, photosystems 1 and 2, taking place in this simulation run. You could also do the same thing with the Calvin cycle, and where glucose gets produced, that's showing carbon fixation. And also just before that is when ATP and NADPH get consumed. So here, ATP and NADPH are both necessary in order for carbon fixation. A second way I use my simulations routinely is just for discovery science. So this is, again, building off of the constructivist idea of having students just play around with the model and seeing results. In order for this to be effective, students have to be willing to perturb the simulation. So here's at initial conditions. Well, if we perturb this by adjusting the distance from light, examining the results. It helps if students write down their results, but at first you might just have them play around and discover what's happening. There's several variables here. I try to make them robust enough so that there's lots of different ways you could use the model. One way is you could gamify it also. So you could say, who can produce 6 grams per mole of glucose during a simulation run? So students will start playing around with different variables and running those variables to see who can make 6 grams per mole of glucose. In order to do that, you have to know a little bit about photosynthesis. So then if students adjust the pH to 8, well, that will increase the amount of ATP because it increases the proton motive force. The hydrogen ions in the thylakoid want to diffuse out because now you have a higher pH, so the hydrogen ions want to move out. That will then have more hydrogen ions go through that ATP synthase, making more ATP. While ATP is necessary for carbon fixation, it's not sufficient. You also need NADPH. So if we increase both the initial pH of the stroma and increase NADPH, well, then we get the amount of glucose at 6 grams per mole, boosting a lot of glucose artificially. Then discussing these results afterwards can be a really interesting process. So then we have this observation of, ooh, what's happening? Why did it do that? Why did Mr. Darko design it this way, and so forth? And leads to this discovery of something that's peculiar, some phenomenon that doesn't seem to make sense, and trying to then work out why that might be. The third way I use my simulations is experimental science. This is hypothesis driven. I have a document for the photosynthesis simulation where I go through like 7 or 8 different hypotheses that students could test with this single simulation. Ideally, they'll start off with some type of claim, and with the claim, then they will collect evidence supporting or rejecting that claim, and then coming up with a reason for that. So here's a couple examples. They could test this hypothesis that the energy source for a phosphorylation of ADP to ATP comes from the electrochemical gradient of hydrogen ions, and they could adjust the amount of pH to kind of test that idea. They could also test the idea that atrazine is an herbicide that affects photosystem 2, not photosystem 1. I will even have students do claim evidence reasoning where they will write a hypothesis and make a little mini poster of the results based off of simulations. Lastly, my fourth way that I frequently use simulations is to recreate features of the simulation. So what I'll have them do is with, for example, the atrazine, have them draw how does photosynthesis, how does the light reaction normally work, draw that, and then draw how it works when you add a bunch of atrazine. They could draw, for example, how varying the amounts of pH affects ATP productions. They could also write the chemical formula for the light-dependent reaction based off of the simulation. So we run the simulation, we might have picked out when the light reaction is, and then just have them draw out that conceptual model of just the light reaction, though, excluding the reactants and products of the Kelvin cycle. That is the main way I use simulations in my classroom with students, try to help them discover how systems work, play around with computational models, and ultimately try to help them construct their own understanding for biological systems. So thanks for supporting my simulations, and we'll see what comes next.